Hi. The title of this video is Why Are They Hiding Your Immune System from You? And we're going to focus on the immune system and why you should not worry about coronavirus even if you get it. In my last video titled Coronavirus Benign, What They're Not Telling You, I focused on why you should not worry about getting coronavirus at all. Basically, to summarize that, and which I summarize with my textbooks, is because, and it's listed as usually asymptomatic, a cold, same family as rhinovirus. Target group is 20 to 40-year-olds, not the elderly or pediatric groups. It's afebrile, meaning no fever. Does not affect the lower respiratory system, therefore no pneumonia. Fastidious, meaning difficult to test for, and on and on. But on this one, like I said, we're going to focus on, even if you get it, why you should not worry about it. Because your own body is equipped with everything it needs to fight this cold virus. Okay, so I have a degree in science, biology, double minor in chemistry, and lots of microbiology courses. My textbooks contain quite a bit on the immune system. That wasn't my, focus in college, but I took the liberty and reread those chapters to refresh my memory. And, so basically what we have is the immune system. We've got specific immunity, which is acquired immunity, and then non-specific. Acquired involves antibodies. Non-specific doesn't even require those. Okay, we've got T cells and B cells. T cells are intracellular, cell-mediated immunity. B cells are extracellular, formation of antibodies, a form of humoral immunity in primary, secondary, and complement. Non-specific immunity involves, it should be an M. Non-specific immunity involves granulocytes and macrophages. Granulocytes digest. Their neutrophils are 90%, basophils and mast cells 1%, in peripheral tissue, not the blood. Lung, mucosa, connective tissue, intestine, and blood vessels. Macrophages are activated by inflammation. Okay, you've got phagocytosis, which engulf the antigen. In this video, an antigen represents a virus, bacteria, toxin, whatever your body is fighting. ATC, macrophages involve ATC, antigen presenting cells. They have a piece of whatever it is, virus, on them, and they present it to the specific immunity. Lymph, skin, spleen, thymus. The MHC codes for self, so that your body doesn't attack itself. And MHC produces interleukin-1. Interleukin-1 is what causes a fever. And then we've got the classic pathway. We've got the complement system that really just magnifies these. And you've got classic pathway, alternative pathway that I'll get to in a minute. But first, now under antibodies, back over here, you've got like IgM, IgG, IgM, IgA, IgD, IgE. Okay. Which briefly, just very briefly, for instance, oops, it's on this page. IgG is the only one to cross placental barrier. Say if you're pregnant, it protects a newborn against infections in which a mother has antibodies. It is a major inactivator of extracellular viruses and bacteria. Blah blah. Okay. Yeah, like IgA is 10% in serum, mostly in external secretions like saliva, tears, colostrum, secretions in intestines, respiratory system. IgG is a monomer, appears on the surface of immune B cells. IgG attaches to mast cells and serves as recognition antibody of human anaphylactic. So that's just very, very brief there. So here's the chart. Let's look at this chart that I made here. Okay, so looking at this, back to the humoral B cells under specific immunity, the primary immune response is when you're first introduced to, say, a virus, okay? Secondary, as compared to secondary, is when you're exposed to it a second or third time, and you've already built up antibodies. The x-axis represents serum antibody level. The y-axis is number of days of exposure. Now, just looking briefly at these two, you can see that the secondary response is much bigger than the first. Okay? The primary and secondary immune responses, in the primary response, IgM is the first to appear in the serum. It appears on the 5th to 7th day. See? The 5th to 7th day. And peaks at about 14 days. Right here. Okay. IgG appears at about the 10th day and peaks several weeks later. Okay. In the secondary immune response, both IgM and IgG appear to increase within 2 to 3 days. So you see, instead of waiting till the 5th or 6th day, you're already there immediately. Bam, it's on it. In the secondary immune response, the lag period is shorter, and total antibody level far surpasses that of the primary immune response. Antibody is formed over a longer period of time, and much more IgG is formed than IgM. IgM here, IgG. IgM, IgG. That's the difference, and that's your immunity. Okay. By the way, I've heard people talk about herd immunity. I found this in my textbook as well. I just want to briefly go over that. That's on page 231 here. I'm reading all this from my textbook. Okay. Herd immunity. When many immune individuals are present in a community, there exists what is called herd immunity. The herd immunity concept is based on the relative number of immune and susceptible individuals in a population. Immune individuals act as a barrier to the spread of infectious agents from infected persons to susceptible individuals. Even though a highly communicable disease may cause an epidemic, many non-immune individuals will be protected because of the unlikelihood of their coming in contact with an infected person. That's just kind of basic common sense. Like, this thing has been going on since, what, last March? I mean, come on. First of all, coronavirus is a winter cold. The heat would have already killed it anyway. But by now, they keep saying, oh, stay apart. By now, everybody, if this was really this epidemic, everybody would have already been exposed to it. You've already probably got, right here, you've already got this. So you've already got this. Antibodies. If you've already got this, antibodies, why do you need a vaccine? Question. Now let's go to vaccines. 177. Okay. Let's go to vaccines. The types of acquired immunity are described below. Remember? Acquired immunity? Naturally acquired. The antigen, usually an infectious agent, are transmitted to the individual under natural circumstances. Exposure to antigen or contact with antigen is unintentional. Okay, so there, you got immunity. 2, artificially acquired. The antigens or antibodies are introduced by artificial means, ordinarily by using a vaccine or antiserum. 3, active. Antibodies are produced by the individual either as a result of an infection or by injected vaccines. Passive. Antibodies are not produced by the individual. Instead, antibodies may have been injected in the form of an antiserum or immune globulins that were obtained from animals or from other humans. The antibodies may have been transmitted under natural circumstances or as a transfer of maternal antibodies across the placenta to the fetus. Blah, blah. Okay, we got on here immunization practices, active immunization. Toxoids, the exotoxins of some bacteria, notably of the tetanus and diphtheria bacteria, are converted in the laboratory to a non-toxigenic form by treatment with formalin or other modalities. 2. Whole cell killed vaccines. Some vaccines consist of suspensions of inactivated intact microorganisms. The whooping cough, typhoid fever, plague vaccines are whole cell vaccines. Whole cell vaccines sometimes produce serious side effects. There are efforts to define attenuated vaccines. Number 3, several effective vaccines consist of living organisms. Now see, before that we talked about killed organisms. Okay, living organisms that through laboratory processing have lost their virulence or their virulence has been reduced. This is called attenuation. Attenuated microorganisms can still multiply in the host, but lack the ability to cause disease. Another advantage, blah blah. Cell-mediated immunity, they include cytotoxic. Okay, the attenuated process commonly involves adapting microorganisms to conditions they do not face in the host. Okay. And then 4, purified antigens, subunit vaccines. Intact microorganisms are a composite of various antigens. The bacterial envelope, for example, has structures or molecular components such as capsin, slime layer, pili. So in other words, they're taking a part of the bacteria and instead of the whole thing and injecting it. Okay, that's just, and, some of these vaccines have side effects. I'm not saying all of them do, and I'm not against vaccines. I'm just saying, why would you, since when do we ever need a vaccine for a cold? Okay. Your body can handle it. Okay. Now, covered that. Vaccines, herd immunity. Another question, okay, back is, Another question. How could each of us produce all these antibodies if there are at the most 1 million genes in the human genome? Bennett said that mutation alone could not account for it. This led to a concept that there are 2 genes for a single protein chain. Or, let's see, it's page 2, or, where is this? Okay, here it is. Or, selective theory proposes that each lymphocyte contains all the genetic information necessary to produce all possible antibodies. Now right there, I had to read that 3 times, it jumped out at me. That to me implies there's a creator of the universe. Okay? Now, let's go back. That's B cells. Let's go to T cells. Let me, go over T cells really quick. T lymphocytes. Their function differ from B cells by receptors on the surface. They produce fast in the thymus, leave thymus, enter lymph nodes to the white part of the spleen. The blood or extravascular space. I had there. You can see that. You can see that diagram there. Whoops. If you can see this diagram here. Okay. You've got thymus, bone marrow, spleen, Breyer's patch, lymph nodes. Okay. There's 3 kinds of T cells. Killer T's, helper T's, suppressor T's. Killer T's bind to the surface of an invading cell, disrupt the membrane, and kill it by altering its intracellular environment. They secrete lymphokines. Blah blah. Helper T's stimulate B lymphocytes, which remember was part of your antibody, to differentiate into antibody producers. T lymphocyte induces also sensitized B cells to divide and mature into plasma cells. Suppressor T's decrease humoral or B cell production so that production of antibodies can be stopped. Okay, did I... Okay, now remember, we've got non-specific immunity involves granulocytes, digest, neutrophils, because it's 90%, basophils and mast cells, 1%, in peripheral tissue, not the blood, lung, mucous, connective tissue, intestine, blood vessels. And the macrophages are activated by inflammation because phagocytosis or engulfing, they engulf physically the whatever antigen, ATC antigen presenting cells. They take a piece of whatever it is, a virus, a piece of it, and present it to the specific community so it can know to attack. MHC codes for self, produces interleukin-1, which causes fever. Okay, let's go to a chart here in the book to show a schematic of this. Let's see, we've got page 164 and 174. Okay. 164. Here we have okay, B cell activation. Okay, this is just basic schematic flow here. We've got activated B cell. Okay, which can interact with a T cell. Okay. Right now we've got B cell. Here's one pathway, proliferation and differentiation. So it goes to a plasma cell and then releases antibodies. Or it can interact with a T cell, and then the T cells do their thing and proliferate. And you got an activated T cell. And then once that happens, the macrophage processing of antigen. That's 174. And 121. No, that was 164. The chart on 174. 12.5. We have a specific immune response. This is an example of a specific immune response in a bacteria. Okay. So, antigen on the antigen presenting cell, APC, makes contact with a T cell via T cell receptor complex. Okay. The activated antigen presenting cell releases interleukin-1, which activates the T cell to release interleukin-2 and increases T cell division. Interleukin-2 increases T cell and phagocytic activity in macrophages. The TH cell also increases interleukin-4 and 6, which stimulate the B cell to differentiate into antibody-producing plasma cells and memory cells. So we got antigen-producing cell, presenting cell. It's got part of the antigen, whether that be virus, bacteria, on the outside. T recognizes it. There's a reaction. And then interleukin-4 and 6 cause B cells to produce memory cells and plasma cells and the antibodies. Memory cells make it remember T cells. So, you see how complicated all this is, right? We've got a, where is that virus thing? Let's see. Non-specific immune response. Here's a non-specific immune response. Okay. Okay. Oh. Okay, so we're gonna, so this is the end of, okay, we're gonna continue this because this is already a 24-minute video. So I'm gonna label this part 1, and then if you just go to part 2, which I'll have in the link below, this will be continued on to complete the rest of the video. We have a lot more to talk about, like testing and the complement system. So please tune in for that.